Hey, all you cool cats and kittens. Welcome to Traits and Reproduction Lesson 2.4, Applying Ideas About Genes. We're going to, as the title suggests, apply some of what we're learning about genes to some different scenarios. We're actually going to go back to actinin 3. Remember that? The runners and the tests that were conducted. We're going to apply some of what we know about genes and gene versions to try to explain what's going on in those scenarios. But first, we got to warm up and check it out. Oh, come on. We got it. Here we go. It's this cool little diagram, and you get to drag these words into the diagram based on where they should go. So here's our words down here: copies of gene versions, feature, gene versions, trait, AUK. So where do all of these fit in the best? Where is it describing traits? Where is it describing gene versions, the feature, copies of the genes? So do that, and then tell me, hey, the spider in the diagram above has homozygous or heterozygous gene versions for the silk flexibility trait. Mr. Reagan, I don't remember what those mean. Or you're going to have to go look them up, aren't you? So do that and come on back to me. Next thing we're doing is this modeling tool activity. So let's go ahead and read our instructions right here. It says, use what you have learned so far to discuss the Chapter 2 question with your partner. You don't have a partner. As you formulate a response, try to use the following words: gene versions, heterozygous, homozygous, traits, and proteins. Hey, you can go ahead and try to explain this to someone you live with. You can email your aunt or uncle and try to explain it to them, or give them a call. Explain it to somebody in your history class. I don't know. But here's our Chapter 2 question. Why do Darwin's bark spiders make different proteins for silk flexibility? Try to come up with a response for that. And then what you're going to be doing is using the modeling tool. So again, in Chapter 2, you have been investigating the relationship between genes and traits. Use the modeling tool, variation in spider offspring model, to show how your current thinking applies to the Darwin's bark spider offspring. Follow the instructions below. Our goal: show why the spider offspring have different traits for silk flexibility. Here's what we need to do. Carefully review the proteins each spider has. Use this information to decide which gene versions they have. Now, in my great compassion for all of you, have actually made this into a Google drawing and attached it to your lesson so that you don't have to go ahead and do that yourself. What you are doing, again, let's go back. Carefully review the proteins each spider has. Use this information to decide which gene versions they have. Okay, so we can see in the cells, if we zoom in a little bit, we can see what proteins they have in the cells. Okay. And what we want to do is we want to make sure we want to say, okay, well, what are the gene versions for each spider? So remember, they can be homozygous, heterozygous. So do they have the same gene version for the proteins that they're producing? Do they have different gene versions? Which gene versions do they have? You actually might want to go jump into the simulation or look at your previous modeling tool to remind yourself which proteins actually go with which genes. So what is the other part of this study? It says add annotations to explain why each spider has the gene versions you recorded and how the gene versions you recorded result in different traits for silk flexibility among the offspring. That's a lot of description there. But do try to add some annotations. You can just add some text boxes there, maybe with some arrows. Here we go. In your annotations, try to use gene versions, heterozygous, homozygous traits, and proteins. So that's a bit of stuff to do. Go ahead, do that, and then come on back to me. All right, next is this, the write and share actinin 3 proteins. So again, we're coming back to this, the experiments that Bay Medical Company did. Here we go. The genetic researchers at Bay Medical Company have sent new data about the actinin 3 protein and the running ability feature. Let's go ahead and read this letter from Dr. Satari together. 2 student researchers, that's you, from Dr. Ada Satari, lead scientist at Bay Medical Company, subject: new data about actinin 3. The genetic researchers at Bay Medical Company are making progress in their research on the actinin 3 protein. Remember, we were trying to decide, does this have anything to do with someone's ability to run? We decided It did. They have identified two commonly known gene versions that help determine the traits for the feature of running ability, A1 and A2. A1 instructs for the actinin 3 protein. This is the protein that helps muscles contract more rapidly so runners can go faster. Use this new information to make predictions about runners in different scenarios. What are those scenarios? Let's take a look. Now, if we were in class and we were breaking up into groups, you would do this with a group. I want you to do it on your own, and I want you to do all three of them just like we did before. So here's the format. We're going to read about the test and then answer these questions. Here's the test, this first one. As you read in Dr. Satari's message, genetic researchers at Bay Medical Company have identified two gene versions for running ability, A1 and A2. A1 instructs for the actinin 3 protein. Researchers tested 3 runners to see what proteins they had in their cells. For the running ability feature, runner 1 had only actinin 3 proteins in her cells. Based on the proteins that were found in runner 1's cells, what can you say about her genes? Explain your ideas using the following words: gene versions, heterozygous, and homozygous. They only had actinin 3 proteins in their cells. So that means they only had, I'm giving you a hint here, the A1 gene. Here's how we would answer that. We would say something along the lines of runner 1 was homozygous for the A1 gene, or runner 1's gene versions were homozygous for the A1 gene. You could say something like that. Yeah, and that's really what we're doing. The whole point here is that we're trying to connect the proteins that they have in their cells and be able to identify their gene version based on the proteins that they have in their cells. Can we identify that gene version? You're basically going to be doing the same thing for this one, except they have different proteins, and this one, and they have different proteins than runner 1. But go ahead, write a response to each of those, and then come on back to me. Last thing, the homework. You're actually going to read 2 articles about scientists that study genes. Before you do that, you're going to answer this. You're revisiting the anticipation guide, and we are revisiting this right here. There are 2 genes that decide each of your traits, and these 2 genes are always exactly alike. So if we go back to the anticipation guide, how did you respond to that? Did you say, yes, I agree with this, or no, I don't agree with this? Hey, you know what? It doesn't matter what you did respond to it before. How do you respond to that now? Do you agree or do you disagree? And here's always the most important part: what evidence supports your ideas about the statement? So tell me whether you agree or disagree, and why do you think that way now? The next thing that you're going to do is you're going to read 2 articles. The first one, Exploring the Human Genome. You can click on this to go there. You can read it right here in Amplify. And then you're going to answer some questions about that. And then you're going to read Meet a Scientist Who Studies How the Environment Affects Our Traits. Again, you're going to go ahead and read this article and answer some questions down here. Now, I feel like that's plenty for you to do today. Here is one of the very, very rare times I am going to go ahead and say, you don't have to do the self-assessment unless you want to. It is actually optional today. Probably one of the only times that it will ever be optional. So if you did watch the video, good for you. You know that you actually don't have to do this part. Anyways, good luck with all that. Looking forward to hearing some of your responses about some of these questions and what you find out. And we will see you next time.